I was asked to talk about two topics. First of all, the genetics of AMD, and then the emerging gene therapy, perhaps where you heard all of, and I'll take you a little bit into the history of gene therapy. When I prepared this talk, I had to, of course, then you go onto Google and you look about Portugal and Netherlands, what's there to be. And then you see, of course, the first 100 pages are only football. So nobody's interested in other things. But I think we have a long way of history there. But perhaps you know that our king and your is that a prime minister? Our president, sorry, are very good friends, and we go a long way back. And of course, a long way ago, we were one of the two most powerful countries in the whole world at the time of the 1600. Let's see here. These were our ships. That were your ships. And of course, this is the 19th century in a nutshell. Of course, you had much more beautiful countries than we had. Whatever. Gene therapy in AMD, that's the first. Genetics in AMD, that's the first. And of course, we all know that AMD is the most common cause of vision loss. And it's a multifactorial disease. That means there are a lot of things that can cause the disease, and the most important of them are, let's go back, are genetic factors and environment. You know that environment are age, smoking, gender, BMI. We go a little bit further. So when developing such a disease, and this is one of the age-related diseases, you have good things. So you can have good DNA, you can have bad DNA, you can have good environment, and here you can have bad environment, like the M of macular degeneration. And when we look in our own files, I have a database of about 4,000 people with AMD, and I collected everything, photographs, OCT, blood, plasma, everything. And we did an analysis, and we do that together with some German groups. And here you see the non-genetic risk factors that are, and of course, is age the most important risk factor. It's an age-related disease. But you said there are some things that cause the disease: smoking, alcohol, gender. Female have a little bit more, but also very important is allergy that seems to protect you from the disease. So again, there are two factors. There are non-genetic factors, and there are genetic factors, and this is one of the age-related diseases with the highest genetic influence. We have also started to look, for example, in DME and other glaucoma. We have analyzed other big data sets in our labs, genetic labs, but nothing is so prominent genetic as AMD. So how is that performed? You have two big data, so you have a big pool, let's say 1,000 patients with AMD, and you have 1,000 controls, and then you take a genetic marker, what we take, and put that on the DNA, and then you will see that in certain areas of the DNA, AMD have more of, for example, a C is a disease-causing DNA particle, and you see it's more in the AMD patients, but it's also there in controls. And when you do this on a big database, and this was the article published by Fritschke in Nature Genetics in 2016, and also we were in that study, and he found that there were 45 common variants, and you have to remember that these are common variants. So these are genetic variants that are very often in in the people, and there are 7 rare variants. That means these are very, very, very seldom there. But the odds ratio, and that's the risks of getting the disease against controls, can vary from 1.5 to 47. And if you divide, so if you take all this and you look at where, for example, this is a complement gene, and you look at all the pathways, surprisingly, you can put them into 3 pathways. And this is the complement pathway, the lipid pathway, and the extracellular matrix, of which the complement pathway is way out the most important. So this is the complement pathway, and then most 99% of the risk factors of AMD are found in the alternative complement pathway. The other pathways are more more influential in bacterial diseases, for example, and this is a complement pathway that gives a slight inflammation, and there are several factors that influence that inflammation. So in a normal state, you have the fact that inflammation is going up or is going down, and this should be a balance. If you get a little bit sick, inflammation goes up. If you're healthy, then it's going down, it's a balance. And due to some genetic alterations, there's a disbalance in the inflammation. But it's only very, very, very slight. So that leads, in the end of the life, it leads to all kinds of complication. And that's why you're not sick in the beginning of the life. Later on, I will show you some examples when you have a very severe affected complement system, then you can get AMD quite early. And you also can measure this in the plasma of patients. This is also some of our patients, and you can see that the complement activation is higher than in controls. So there are 3 important risk factors: the complement pathway, the lipid pathway, and the extracellular matrix. I just give you now the genetic factors that lead from a normal fundus to early AMD to intermediate AMD and eventually to geographic atrophy or choroidal neovascularization. So what we saw until now are the common variants, as I was talking, that explain a lot of the AMD, but they're very common and they have some effect, but on very later life. Also, we know that there are families that are full of AMD. I don't know if you have them, but I have about 75 families collected where more than 5 people have AMD. And these cannot only explained by the common variants. There should be rare variants that have a strong effect. They're really like mutations, like inherited diseases. Here you see some of these examples of these families. And we looked closely into these families. And then we found in the same genes as that play a role in the majority of AMD, we found these mutations. And you see the odds ratio is very high. In these patients have, for example, with this mutation, have a risk that is 20-fold more than controls to get AMD. So that's very, very, very strong. Nearly all these patients who have these variants can get AMD, whatever they do. This is, for example, a family with such a mutation. They have a mutation in the CFH gene. That is the gene where normally the polymorphism, so the slight genetic alteration plays a role, but this is the strong genetic alteration. And you see they're also relatively young for getting drusen, but also if you look at the younger patients, you can find drusen in the periphery. So if you find drusen in the periphery, especially when you find a lot of drusen in the periphery, you have to look for these very strong variants. And why I'm telling this? Well, where does the complement system, is there a therapy? Now, what was very interesting is that, and this came from the veterinarian sides. So they give, in the winter, they give horses zinc that they get less sick. And we already knew somewhere from literature that there was an influence of zinc on the complement system. Zinc is a complement inhibitor. So we tested that in patients. We took patients with a high complement activation, so patients with mutations or with polymorphisms in their complement system, and gave them zinc, and then measured the serum complement activation. And you see it's going down. And when you stop zinc, the complement activation is going up. However, if patients with AMD do not have more complement activation, zinc doesn't do anything on the complement activation. So that could also explain why complement inhibitors like lampalizumab didn't work on the whole group, because complement suppression only works in a certain amount of patients. And here you see Emily Chu and the ARIDS-2 study, and complement, zinc was the main factor that worked in not getting AMD, in the prevention of getting AMD in people with high risk. So probably the working mechanism of zinc is the complement system, and that brings us back to genetics. So now we understand complement is a strong genetic factor. Zinc works on the complement system, and complement inhibition by zinc can be explained by suppression of the complement system. So this gives us insight about the disease. So how can you see it? I see AMD as a card game. Genetics you cannot do anything about. If you want, you cannot change your genetic profile. So the genetics gives you the cards in life, and the environment plays the game. So if you have very good cards, if you have very good genes, and you play the game very badly, that means you smoke and you drink and you do everything, you still can win. You still get no AMD. If you have very bad cards, so if you have mutations, you can play the game as good as you want, you always lose. So that's a little bit how these disease develops. So remember, it's a card game, genetics gives you the cards, environmental plays the game. That's the way. And we want to treat here. But should we treat here? For example, in dry AMD, we want to treat there. We want to treat in intermediate AMD, or even in early AMD. But should we treat all these people only because these people get the disease? No, we want to know the influence of genetics and environment on the development of late AMD. A lot of people thought about that, and people develop all kinds of complement inhibitors, you see, and they do not work. They do not work because it's a bad idea, but they treat the wrong group. And you see, targeting the wrong components, treating too late in the disease, and selecting patients on basis of genotype. Should we not look at the genotype, see the patients that have a bad genotype, and then treat them with complement inhibitors? So we want to treat them there. But then we have to know what are the people that get the disease. And fortunately, the European Union gave us money to investigate that. And that's the MacuStar Consortium. And you see Portugal is there, and we work nicely together. Portugal and the Netherlands, Coimbra is there, and Nijmegen is there. That's where I'm from. And there's also another consortium, IRIS consortium, where we look at making or producing a predictive test in AMD. So that's the use of the genes. This is just the beginning of the story, but I think on hand of these genetic knowledge, therapies will be developed in the future. Okay, that was genetics in AMD. Now we have to change to a whole other subject, and that is gene therapy in retinal dystrophies. I think you are all genetic specialists, so you have all dystrophies in your practice. And I'm a medical retina specialist, and I do both AMD and ophthalmogenetics. But ophthalmogenetics is getting more and more difficult. Why? Because there are so many genes. I mean, this is what you have to know if you want to do proper ophthalmogenetics. You have to know all the genes, you have to know how they work, you have to know what is the protein, what is the defect on the protein, and all the diseases they can produce. So, and you see there's a lot of overlap. So some genes, like before the ABCA4, which is one of the most prominent genes, that can give RP-like thing, it can give rod cone, but even perhaps it plays a role in macular degeneration. We are just publishing a few papers, and if you read them, just wait half a year, they will come out. I think we have found a new role of ABCA4 in age-related degeneration. So I'm only talking today about gene therapy. So the gene therapy that has been developed until now. There are now a lot of other therapeutic strategies. For example, RNA modulation. We cannot only try to fix the DNA, which is difficult, which has disadvantages. I will talk about that later. But you could also try to repair the RNA. The RNA is outside the cell, so you can stay outside the cell. Or you can even work on the protein. Try to bring the protein in. So, but that would take a whole day to talk about that. So if there is a mutation in the DNA, you have a defective RNA and eventually no protein or a little bit of protein is made. Depending on how severe the mutation is, the RNA is affected and the protein is affected. If the mutation is very, very severe, you have no protein, the cell won't function, and it will lead, for example, to blindness after birth. If the mutation is very slightly, and we saw that in AMD, you can get the disease at a very old age. So what's the therapy? Very, very easy. Everybody could thought about just take the gene and bring it into the cell. Case closed, patient is healthy, can see again. That's not so easy. And you make new protein. So I will give a little bit talk about the RP65. That is now the first gene therapy that is available on the market. So this is the history. We know that the DNA was invented and found by Crick and Watson in 1953, and DNA sequencing was available from 1977. The PCR was invented, was also the Nobel Prize at that time. In 2001, we knew the whole human genome, we thought, but now we know there's a lot more. And you see here are the first gene therapies coming in after the late 80s in the 90s. At first, for example, 1990 was the first human gene therapy trial. So when we could already do trials in 1990, why took it so long to get the first compound now on the market? Now that's quite difficult. This was first Klein experiment about gene therapy, and that was the first human gene therapy, and this was published as the first gene therapy, but I will get to that later. But this is where I want to talk a little bit more about this slide. That's why I skipped slowly others. Then there was gene therapy, and of course they started gene therapy for very severe diseases. So this was a boy who had a very severe disease, and he was going to die, and they did gene therapy. It was an inherited disease, and they did a gene therapy with an adenovirus directed to the porta vein, and the boy died 4 days later due to the gene therapy. And this put the whole gene therapy development to a halt, because somebody had done a trial where a patient died. And what were the problems in this study? Nobody had really decently looked at the toxicity of the study. So they were going right into humans. There was a big financial conflict of interest of the PIs, which made them try to do the trial quicker than with decent studies about tox studies and studies on safety. And the subject did not meet the inclusion criteria, and there was even later no informed consent. So then the government in the United States put the whole gene therapy development on hold. Until 2007, there was in ophthalmology the first RPE65 trial. And why could we go in ophthalmology relatively quick into a human trial? That was because, sorry, I'm skipping a little bit. I wanted to tell you that there was a dog. So normally you do studies in mice, and first in the lab, and then you go into mice, and then you do a lot of studies. But the mice eye, the eye of the mouse, is something completely different than the eye of a human. So especially in inherited diseases and in gene replacement, the mouse is not very doesn't look the same as a human, so that is very difficult. And this first disease was about Leber congenital amaurosis. And of course you all know that is a retinal dystrophy where you get blind in the first years of your life. So these are all the genes that can cause LCA. By the way, I do not like, we just have published a paper in Prayer where we say LCA is just a bad name. So where's the border between LCA and RP? I think this is all RP. It's just early onset RP, because these are early. Because, for example, we have families with TOP mutations where some kids are affected very young, but some adults are affected much older. So it can be all. This is RP65, and the gene is there. It's in the retinoid cycle, outside in the pigment epithelium, and converting to L-transretinol to L-cis-retinol. And this is the defective gene, and that's why the photoreceptors go away. These are the mutations that are found in that gene, and these all lead to loss of function. So what would be an idea? Just grab the gene, inject it into the eye, and put it into the nucleus. So this is how that's produced. You take a virus, And why do you need a virus? Because a virus normally infects a cell. So a virus is nothing more than a small envelope, and with the possibility to infect a cell, it can inject its DNA material into the nucleus, and then it produces new sickness genes, like in herpes, like in flu, but also like in HIV, whatever, all these viral infections. So what you do, you use the capacity of the virus to infect cells, but you just exchange the DNA. So you take out the bad DNA, which makes you sick, and put in the gene which is missing in the patient. And of course, in an eye, we have the advantage that we do not have to inject it into the blood. So it will not infect the whole body, it will stay in the eye, but only the retina cells are infected. So you change the gene, this is the normal AAV, this is the replication gene and the capsid gene, and you put this instead and put it into a virus, and luckily it stays infecting the cells. And this was a dog in Germany, Andreas Gall, of Sweden and Germany, Hamburg and Sweden. They found a dog that had this disease which the human had. Normally, it would be ideal if we would have dogs with blindness that have the same blindness as human people. But unfortunately, until now, we're throwing the dogs away. So if a dog gets blind, I mean, you never see blind animals, they get eaten or thrown away. We don't put them in the institute. So that's why we miss these perfect animals for research because they don't survive. Now there's a big European program to save blind dogs. So of course, most blinding diseases are recessive. So there should be dogs that get blind. Logically, there should be dogs that get blind, but they are thrown away. So now we try to save them and see if they have the same disease as the humans, and we can use them in research for gene therapy. So this dog had the mutation, and perhaps you have seen, I didn't bring the video, and they injected this in the eye of the dog. And this is about how gene therapy, I'm thankful for my colleagues in Tübingen who allowed me to show this video, and this is how gene therapy is carried out. Because the retina has still the ELM, the internal limiting membrane, the viruses cannot get through there. So you have to inject it under the retina, which is still a vitrectomy is needed. So can you see it a little bit? Yes, sir. So now the virus is injected under the retina. Mostly it's injected above the macula, and then you make a big blab and you put the patient sitting in a chair, and the fluid will go down under the macula. And after a day or 4 or 5, the macula is attached again. So these are the phases of the clinical trial. First we had to do, not we, it was the group in the United States, had to do safety, efficacy, and the phase 3, the approval studies. So this was injected, I was to see, yes. And under the retina, this was how it was done. This was the gene they made. You see here where they inject, you see it's injected above and it goes down there. These are all the patients. There were 2 studies going on. McGuire and Brainbridge did the studies, all published in the New England Journal. And you see that especially the patients in the United States improved of visual, from hand movements to 20/30. But especially because these are so low visions, you want to test, and potentially this therapy is so expensive, the authorities want to know if the patients can live better or have a better performance in daily life. And like I say, life is not a letter chart, but life is life. And you want to know, after such an expensive procedure, you can perform better in life. So this is, they make a maze, what they call. So a circuit. Could we have the sound? You can see that it's written there. This is the eye that did not have surgery. And in the near future, we are going to do gene therapy also, and now there's an official maze, which is 4 meters by 7 meters, and you can make all kinds.
É possível pôr o som no vídeo?
At least I can see the black squares.
Okay, stop. Okay. Turn right.
Those are.
So this is he had light perception in both eyes. This is the eye that has had no surgery.
We can't make it too easy for you.
Well, the best you can. Anything. Just do the best you can. Again. Again, the stop. This is being really hard.
You see, he cannot find his way and move perhaps. And now the other eye, after surgery. This is 1 week after surgery.
And you can start whenever you're ready.
Okay, so the gene therapy RP65 appears to be safe and moderately efficient. Younger subjects should benefit more. We do not know yet the long-term benefits. And RP65, of course, had all the advantage. They had the dog model. That's why they could move so quick into patients. And still, you need 8 years of safety after doing a trial. So after doing a human trial, you have to wait for 8 years to get a registration of the drug. And these are the gene therapies at the moment in research. There's a study on Stargardt, but that's on hold now. There's achromatopsia, choremia, done in Oxford by McLaren, and we are part of that group. Usher 1B and X-linked retinitis pigmentosa, also by the Oxford group developed. And so there are several trials out. Choremia is now also in its phase 3, so this is now after RP65 that is now marketed by Novartis. Choremia will be the next player on the market if efficacy is good enough in phase 3. Okay. One thing to remember is that the price is high. There are only a few patients. The development of such a drug, you see, takes years and years and years and years. So the costs are quite high for one treatment. Fortunately, there are other developments that will, so for example, you could repair the RNA. There are studies on bringing proteins in, repair of the RNA, but that's the future. I can tell you perhaps at another time, that's a very interesting field. But until now, I would like to thank you.